ready to learn?
Always.
Okay. So as you all know, there's another exam coming. So 11:18, I moved the exam because, as you know, everything's been off by a week because I'm adjusting to teaching twice a week. So 11:18 is our next exam. I'm sorry to bring up the word exam since we just took one, but you should all be very happy because people did really well on this exam. So, and I'll show you the distribution in a minute. Some quick business. Stump the teacher. Someone asked last week about, I think, was it Stephen? Was it you? About whether or not motor proteins and microtubules were involved in the movement of the SRP to the ER. And I didn't think so, but I wasn't sure, so we made it a stump the teacher. And I got a bunch of people who emailed me, mostly saying no, it's not, there's no evidence. And that's really good because that's what I thought, and I don't know why there would have been any evidence for movement of SRP by motor proteins, because it's usually more involved in movement of vesicles or organelles. SRP is just a protein complex with ribosome, which is huge, but yeah. So that's where we got. That's where we got with that. Some people emailed me and were like, "Why didn't?" Or came and spoke to me and were like, "Why didn't tell you anything because I couldn't find anything." And I was like, "Well, usually when you can't find any information about something, it might be because there's this huge bias in the literature where people don't publish negative data. No one's going to write a paper that says motor proteins do not move SRP to the ER. So if you can't find anything, it usually means maybe it doesn't happen. I don't know. You can always email me and say I have found no evidence." So those of you who sent me something that is along the lines of what I just said, will get a point for your efforts. Okay. Another thing, critical concentration and treadmilling. We had a little bit of trouble with that on Wednesday, where people were asking me all sorts of questions. I got a little confused. So we're gonna attempt it again shortly, and you guys will let me know how it goes. Okay. Exam 2, average is 83. 83, amazing. Which means I'm too easy, probably. So we're gonna bring that down, back down to 75. No, you guys did really well, and I think it may have had to do with that last review session, is my theory, because people were on it. So, again, if you were down at this, under 60, some 65, please see me. A number of people have seen me, but please see me. Okay, so far, this is what we have done. We've only had one lecture on cytoskeleton so far, so we're still in our introductory phase. But we spoke about the key players of the cytoskeleton, actin, microtubules, intermediate filaments, and I mentioned accessory proteins. I haven't told you much about them yet, but we're going to dive into them soon. And then on this last weekend's quiz, you guys had to do some homework learning about the accessory proteins. Then we started talking about some major concepts that we need to understand in order to talk about the cytoskeleton. We spoke about dynamic instability, the fact that filaments or polymers are produced from subunits or monomers, the concept of nucleation, the concept of critical concentration, the fact that these filaments have plus ends where they're more apt to grow and minus ends where they're more apt to disassemble, and treadmilling. And then we started talking specifically about microtubules. They're made from alpha beta subunits. GTP is bound by by tubulin subunits and hydrolyzed upon addition to the microtubules. The GTP cap determines growth versus disassembly. And then we started talking about actin and how it's similar in that there's a nucleotide, in this case ATP, which is hydrolyzed after addition to the growing actin filament. And that's kind of where we stop on Wednesday. And so as always, if you keep to this list that keeps growing every lecture, you should know what will be on the exam. And then someone was asking me if I was going to do a review session the same way we did it for the last one, and I'll see if we have time, but you guys can start doing that as we start to learn about the different elements of the cytoskeleton, mostly actin and microtubules. Draw it out, and as we talk about all the different things that happen and the different accessory proteins that regulate assembly or disassembly or nucleation or whatever, add them in and make a little note as to what they're doing. And so you can kind of start to create this monster drawing that will have everything in it. Okay. So here's another concept, which I think that I had mentioned, but this is where we're going to start. So ATP and GTP is hydrolyzed when the subunit assembles into a polymer. And this kind of regulates the binding in a way. So when you add your subunit and it's in its T-bound state, so that's referring to ATP or GTP, it's going to bind more strongly than when it's in its D-bound state, so after hydrolysis. And so that explains why with the microtubules, when you lose your GTP cap, you start to get disassembly. So hydrolysis reduces the binding affinity of the subunit to the neighboring subunits, making it more likely to dissociate. So the T-bound monomer will add to the filament, and the D-bound form is more likely to leave. So let's just go back to critical concentration for a minute. So the definition, or my definition at least, is it's a concentration. We're talking about a concentration of free subunits in solution. And when the rate of addition of new subunits is equal to the rate of dissociation, you're in this steady state. That's the critical concentration. Okay? So it's an actual concentration of free subunits in solution. Okay? And so it has units, because it's a concentration, which are micromolar. And the critical concentration is different for the plus n or the minus n. And so that, I think, after class, when Sam was asking me a million questions about this, we realized that something I had left out the other day was explaining that each end of your growing polymer has its own critical concentration. So it's not to say there are different concentrations of molecules at each end. It's that their critical concentration is different for each end, and that kind of makes sense when you consider the fact that you're going to add differently to your plus end and to your minus end. So your critical concentration is greater for your minus end, and it's lower for your plus end. So we're going to show you some pictures that will hopefully help this. Okay, so remember, we've got our subunits, which are in our T-bound form, ATP or GTP, and your polymers are going to be a mix of T and D-bound form, right? So here's your subunits and the green squares, and so they're being added to your polymer. And so remember, you're adding more rapidly to your plus end, so you're going to have a buildup of your T-bound form, and more slowly to your minus end. And because of that, remember, the hydrolysis of your ATP or your GTP is going to eventually catch up. So since your plus end, you're having this rapid addition, hydrolysis lags behind, right? So you're going to have a slower addition at your minus end, and your hydrolysis catches up, so you have more D-bound form. Yes?
Why does the hydrolysis lag behind the catch up? Why would it?
Well, so why does the hydrolysis lag behind for here? So
Yeah.
so when you add your monomer to your polymer, you get hydrolysis. Okay?
Right.
But if your critical concentration is fairly low, and so you actually have a high concentration of monomer, so you're adding quickly. You're adding so rapidly that you add faster than hydrolysis can occur.
Okay.
Okay?
Yeah.
Okay. Did people catch that? Yes.
So I just want to be clear, on the last slide, you said that when the rate of, or critical conditions, when the rate of addition of new subunits is equal to rate of dissociation, but each end is gonna have its own rates that will balance each other. So, I know it sounds weird when I say it, but that's.
Yeah, so this is going back to Sam's question the other day. So when the rate of addition of new subunits is equal to the rate of dissociation, we're talking about at one side, at each side. And so it's this steady state, so you get addition, removal. So if you're just looking at the plus end, you get addition, removal, addition, removal. So you're not going to get any growth or shrinkage. You're at the steady state, that is your critical concentration. When the concentration of monomer is such that you're not getting growth or shrinkage, it's just equal addition, equal removal for each side separately. Okay? Are people fine? I'm seeing a few people going, and that's good. That's good. Okay, I think we're gonna have a video. I love movies. Yes.
What is the mechanism that makes the plus and minus n different? Why does it add faster at the plus end than it does at the minus end? What distinguishes
What distinguishes the plus end from the minus end? What distinguishes the plus? It's okay. So the plus end is the plus end because of that rapid addition at that end, and because so you can choose your plus and minus end. Like so in this picture, it's showing to the left or to the right. Obviously, it has nothing to do with it, but always there's this polarity of the addition where you're going to have each time you add, you're going to have hydrolysis, right? You're going to have your T-bound, and then it's going to hydrolyze. You're going to have your T-bound, and then it's going to hydrolyze. It's going to turn to its D-bound. The side that has your T-bound is going to be your plus end. So that's where you're having your more rapid addition of your T-bound monomer. And the side where it's older and it's been around longer and there's not as much addition, that's your D-bound, and that's going to be your minus end. So it's more is not that there's an identity. It's more it has to do with where the subunits are being added.
Why are they added at that end rapid?
Why are they added at that end rapidly? How can I explain this better? Yes.
Because there's more free subunits at that end?
Well, it has to do with, see, now I'm going to get myself twisted again. Is it because there's more free subunits at that end? Well, so it's not that, because you're in a cell, and there's a concentration of monomer in the whole cell, right? But, let me think of how to answer this. Oh, I feel like I have students who can answer. Yes?
I'm wondering, does it have anything to do with like your physical level last week with this like forming or the other?
Okay, so I'm getting questions about accessory proteins. Let's talk about actin and tubulin just in a test tube without accessory proteins. Yes.
Is it the polarity of the subunits?
Right. So when you add, so I wonder if I can go back. No, let me go forward. So the polarity of the subunits. Yes, it has to do with the polarity of the subunits. So when you're adding, so you have your who's asking me questions? Everyone's asking me. So when you have your tubulin dimers, you're going to have your beta tubulin has your GTP, and that's going to be hydrolyzed, and it's added in that orientation. So in a sense, yes, it does have to do with the orientation in which they're added. So you're going to have so there's your alpha beta. Oops, that's a dimer. Right, and so this is where your GTP is going to be hydrolyzed. So it's always added in that order, so you're going to, I don't know if that, does that, someone else was, Raven?
I was gonna ask if it has to do with
Does it have to do the, well, in terms of, one is going to hydrolyze the GTP, and so the binding, so the interaction between the alpha unit and the next beta subunit is going to be affected by whether you're not your GTP bound or GDP bound. So yes. Any other questions? Yes?
Would it have anything to do with like that T form, D form? Like if there's more T at the plasma, then it's gonna bind more
Right,
readily there,
right. So, okay, so the T form, D form, right, so the T form binds more strongly, the D form dissociates more readily. So you're going to add more readily at your GTP cap, where you're having more T form. So you're adding more and more quickly, and that defines the plus end. Does that make sense?
Yes.
Okay. Any other questions? Okay, yes.
Just to clarify, just something you said, so the rate of increased addition on the plus side?
So the rate, so where you're adding more quickly is by definition your plus end.
So then you switch depending on size being added?
Can it switch? I don't know, because really it's kind of controlled by your rate of addition, and so something would have to, yes, why not? Accessory proteins. When we talk about accessory proteins, you'll see proteins that can force disassembly or can force growth. So let's revisit that question when we get to accessory proteins. Okay. More questions. We're on a roll. Okay, let's just keep moving and see what happens. Okay. That's where we were. Okay, so treadmilling occurs at concentrations above the plus end critical concentration and below the minus end critical concentration. So remember, each end, your plus end has its own critical concentration, because remember, it adds subunits at a different rate than your minus end. And so if you look at this graph, and so this is your elongation rate. Here's 0, here's your steady state, and so when you cross that, that's your critical concentration. When you cross that line, you're growing, and when you go below that line, you're shrinking. So as your subunits increase, if you look at your plus end, as your subunits increase, once you cross your critical concentration, you're having growth, right? So you're above your critical concentration, you're growing. Here's your minus end. When you're above your critical concentration, remember this is your concentration here, you're growing. And so if you're below your critical concentration for your minus end and you're above your critical concentration for your plus end, you're treadmilling. And so I'm going to show you a movie, which is nice, in just a second, that will help with this. Okay? Good? We want to think on it for a minute? Yeah. Okay. So treadmilling is resulting from this net assembly at the plus end and a net disassembly at your minus end. Okay? So if you kind of you can kind of follow your red here, right? So it's these same subunits. So you've got this net assembly, so it's growing. So it's growing, and these are the same ones that are moving this way, and then they're going to come off. So the polymer is maintaining a constant length, but you have this flux, this net flux of the same subunits through the polymer. So if you look at this figure here, what they did is they labeled tubulin in a microtubule, and then they did time-lapse photography, and this is right out of the textbook. So at time 0, you can see the green here. At time 0, it's here, and then after 98 seconds, it looks like it's here, and it's not actually moving. You just have this growth at the plus end combined with the disassembly at the minus end, which makes it look like it's moving. Okay? Yeah?
So it's moving kind of like a bicycle chain.
It's moving like a bicycle chain. I don't know about that. Because really it's not like, so are you thinking it's coming, the same subunits coming off one end and adding to the other end? That's not necessarily the case, because they're just coming off, and they're just adding, they're not necessarily transiting to the other end. So, but that's an interesting way to think about it. Okay, how are we doing? Yes? If the microtubule is not actually moving, It is moving. So the microtubule itself is moving, but it's not like it's crawling. You know what I'm saying? So when we start to talk about applying this, when we start to talk about cell movement, that's my favorite one with actin. Because you have treadmilling of actin, and so when the cell is moving, you actually have this actin growth at the plus end and disassembly at the minus end, and that actually pushes up against the plasma membrane of the cell and pushes the cell forward, and that is kind of a treadmilling behavior, right? Huh? Is it just for moving? No, so in this case, this is just here it's not so much that we're moving. With organelles and vesicles, we have motor proteins that are attached to the microtubules that are walking along and moving things. This is really just to understand how there's disassembly and movement of microtubules and actin filaments in the cell. Yes.
So this is like when back in the day when they were moving really big things, like the really big bricks, and they had the logs. They had like 20 logs, but they're not the same logs. They could be different logs. And the people are running around to the front, so it's moving and it stays the same length, and it's moving something, but it's not necessarily the same log that's going to the front of the track.
I kind of am following you, like a bucket brigade.
Kind of. But like it's the same length, but it's not the same substance building. And it's moving something along in a forward direction. Same length.
Yeah. Yep.
Whatever works. Whatever works.
I'm open to all analogies. Let's watch a video. It's like a log bucket brigade. That's like kind of how I'm seeing it. Yes.
Can I ask a question about this? So where the red arrow is, if we're thinking about that in terms of like position, that person they're at that position, whatever's there is staying at the same place, but then it's just being added or subtracted, and that's what we're seeing. Is that what the red line is?
So the red is, yeah, so let's see. So here it's showing either end, and so here it's, I think you're following one spot on the microtubules.
So that's staying in the same
And that's-
place throughout the same time. Is that what that's showing?
I believe so, but we have to look at the figure legend in the book. Okay. Movie? Okay, we're gonna watch it a bit. Okay, so we're gonna watch it a few times because that's really fast. But so you can see your ADP actin is most likely to leave, that's your yellow, and your ATP actin is most likely to add, that's your red. Let's do it again. That's treadmilling. So you're adding quickly in the front and disassembling quickly from behind. And you're staying, I'm doing it again, you're staying the same length, generally. Can I do it again? Treadmilling. I know. I should always do the visual first, shouldn't I? Okay. Okay, so did that, yes.
Okay, so.
Do I see it again?
Something again, but if this is right, this will completely answer my question. So, the treadmilling, and the critical concentration. The critical concentration, having part of that be the addition and disassembly. Is the plus side the addition and the minus side disassembly?
Well, no, so, okay. No. So when we talk about critical concentration, we're talking about the plus end, addition, and disassembly. Minus end, addition, and disassembly. Because you're adding and disassembling at either end. And it's just where you are in relation to the critical concentration, which is going to determine whether you're going to grow or disassemble and shrink. So if you're at the plus end and you're above the critical concentration, you're adding. But if you're below the critical concentration, you're leaving. So I feel like we need to move on for a second and come back. Because there's some magic thing that it's been 3 years I've been teaching this, and I still feel like there's some magical movie that's going to help it more easily. I went to ask a few of the A&P folks, because I was like, they're going to know this, and that people who are teaching advanced phys. I'm like, so what do you guys think? How should I teach this? And they're like, oh no, we don't go that deep. And I was like, maybe that's my problem. But you guys can do it. Okay. So let's go on. Okay. Microtubules. Okay, so growth and shrinkage is dependent on the presence of a GTP cap. I keep talking about this GTP cap. And that's because, remember, as you're adding at your plus end, you're going to get this rapid addition, which results in this buildup of the GTP cap. And the fact that it is GTP bound encourages further addition. But when you drop below your critical concentration, there are no more monomers to add. And so then that GTP hydrolysis can catch up, which results in disassembly. So this is what you're seeing here. So you've got your GTP cap, you get growth, you're adding more, and then you start to lose your GTP cap because you're not adding any more monomers, and that's called catastrophe. I love the fact that it's called catastrophe. You'll never forget catastrophe. I've never forgotten it. And it's because it is. It's just like the complete disassembly of microtubules. And so that results in this rapid shrinkage. And don't forget, when you get the shrinkage, all this monomer is leaving your microtubule and going into solution, which creates a higher concentration in solution, and then you can start getting addition again, which results in rescue, and then you regain your GTP cap. And so that's what's constantly happening. You have this disassembly, disassembly, so monomers go into solution, and then you have reassembly until you drop below your critical concentration, so you can't grow anymore. Your GTP hydrolysis catches up, and then you get catastrophe, and again and again. And the only thing to keep in mind, this all seems very abstract right now, but as we start to talk about all the different processes that this is involved with in the cell, and we talk about the accessory proteins that help to control this, we can start to see why we should care. I kind of tried to make you care about this the other day with all these movies, so we're going to tie it all back in. Okay, so but this is dynamic instability. It's constant growth and shrinkage. Okay. So another way to think about dynamic instability, the GTP cap keeps microtubules growing, and then when that cap is lost through GTP hydrolysis, depolymerization is likely to occur. So I'm talking about this so much because they're just important concepts to understand just the general function of the cytoskeleton. This treadmilling and dynamic instability, this is what helps with the aid in the rearrangement of the cytoskeleton. You need to, when you're going from interphase to mitosis, you need to disassemble your microtubules, and you duplicate your centrosomes, and you're going to make them into your spindle folds, and you're going to grow your microtubules and center them, and you need to be able to disassemble and reassemble and control all of this. So actin and microtubules, they switch between growth and shrinkage every few seconds for actin to minutes for tubulin. So this is rapid. This is truly dynamic instability. So the cell has to use a lot of this NTP to keep this constant concentration of filaments. So it seems like a waste of energy, right? You're constantly, each time you add to a growing filament, you then hydrolyze your GTP or ATP. Seems like a waste of energy, doesn't it? What's the advantage to this constant addition and removal? Addition and removal, yeah.
NTP.
NTP, yes. NTP just means nucleoside triphosphate because remember, microtubules use GTP. Thank you for pointing that out. Microtubules use GTP and actin uses ATP, and so sometimes rather than writing everything, I just say NTP, nucleoside triphosphate. Yep. So what's the advantage? It seems like such a waste to use all this energy. What's the advantage of this constant rebuilding, disassembly, rebuilding, disassembly? Why do we want to do this? Yeah. So it allows the cell to adapt to its environment, which is constantly changing. There's always something going on, something new that's happening. Anyone want to add to that? How about from this end of the room?
Yeah.
So does it have something to do with cell movement and mitosis? Well, yeah, absolutely. So that's, you're adapting to your changing environment because you might need to be a neutrophil that needs to chase bacteria. Or it might be time to go through mitosis. So you're going to have to disassemble and reassemble. If you imagine that all you're doing is breaking things irreversibly and creating these permanent structures, it seems that seems even more wasteful than just this constant hydrolysis of NTP, right? So the constant turnover is providing flexibility in reorganization of the cytoskeleton to meet the needs of the cell. Did you read the textbook? That's almost exactly what you said. So if you look at this, so this is just microtubules over time, so 0 through 669 seconds. That's a strange number that they chose. And so you can see they're following each one. So you've got 1, 2, 3, 4, and you can see that this one's here, and then it's down here, and then it's up here, and then it's down here. And if you look at 3, 3 is short, then it's long, then it's shorter, then it's shorter. So you're just constant change, and that allows for adaptation to the environment. And so these subunits are small, and they can rapidly diffuse through the cell. And then don't forget all these accessory proteins help to recruit proteins. So remember when we spoke about, now everyone has done recitation last week, and we did that paper on E. coli and pedestal formation, and how E. coli recruits ARP and WASP, and that promotes actin polymerization to create this pedestal for tight interaction, right? So there are all these actin subunits in the cell, and E. coli, by recruiting WASP and ARP, which are two of these accessory proteins that promote actin filament polymerization, it's forcing the cell to change its conformation, to create this pedestal structure. Okay? How are we doing today? Yeah? Is everyone happy? Do we have a good weekend? Okay. That's good. It was nice out, then it got cold. I tailgated. I've never really tailgated before. I went to see the Tigers played the Delaware. I don't know. I didn't go to the game. I didn't go to the game. There was a birthday party there that I went to in the Graf's house. Okay, this has nothing to do with cytoskeleton. Though my cytoskeleton probably had to adapt to how cold it was. I don't know. Okay. Okay, so let's just think for a minute. How might actin and microtubule activity be controlled differently? So we're thinking about actin and microtubules, and their building, and their disassembly, and this and that. How might they be controlled differently in different types of cells? In a neuron versus a macrophage, what controls these differences? So I want everyone to take a minute to talk to their friend about this. And just think to yourself, what are the differences in these cell types? How might actin and microtubules be controlled differently? We'll take a minute and then we'll discuss. Okay? Everyone can have a brain read. Go.
What?
Okay, okay. Okay, guys, so we're talking about how actin and microtubule activity might be different or controlled differently in different types of cells. So you're comparing a neuron and an macrophage. So let's first talk about actin. How do you think it might be controlled differently in a neuron versus a macrophage? So think about the jobs. I heard a lot of talking. Yes. Yes, no.
Beer.
Oh no. Okay, everyone raise your hand. Go ahead.
Well, macrophage, they're a lot more active moving around. So some would be more rapid and more other, but the neurons are pretty static. Once it's made, it's made. There gets to be a lot more activity in macrophage than neurons as far as acting in macrophage.
Okay, so what I'm hearing is macrophages move more than neurons. Does everyone agree? Yes. Do neurons move a lot? No. So we talked a little bit about how actin drives movement, actin polymerization drives movement. So you'll probably have more actin assembly, actin polymerization in a macrophage versus a neuron. Okay. So what about microtubules? A little bit more difficult. You have to think about what microtubules do. Should I get my class list out? Bailey.
They have more distance, like because the axon is long. It's got more distance to that travel, so it probably has more activity with microtubules or the transport.
So neurons, right? So neurons have a long axon, and so they have more distance that needs to be traveled. So how do you think that might affect microtubules? Do you think they'll assemble, be more stable, or disassemble more rapidly?
Well, both. Because I mean, they would need the stability if it was constant movement, but if it only needs to move something in the beginning of it being made or the very end or something like that, then it wouldn't necessarily need to be incredibly stable. Depends on the traffic that it has.
So it depends on the traffic, yes.
Do microtubules have to do anything with the channels of neurons?
Do microtubules have anything to do with the channels in neurons? So like neurotransmitter secretion? Is it called secretion?
Yeah.
Yeah, because so remember vesicles, secretory vesicles and stuff like that are going to be moved along microtubules? Yes, do you have?
Yeah, I was thinking more of like how actin filaments play a big role in neurons in terms of maintaining cell shape, to maintain the synapses in the proper position.
Oh, so actin filaments might be more stable, maybe to maintain synapses in the proper position? Maybe, I wasn't thinking about that. Maybe. Yeah. Anyone else? So this just speaks to the fact that you want to have this ability to reorganize rapidly in response to the cell-specific environment. And so what's going to happen in a macrophage that's near bacteria is going to be very different from what's going to happen in a neuron that's signaling. Okay. So what controls these differences? Now I'm going to make you think deeply. What controls differences between cells? Yeah.
Doesn't it depend on which genes are expressed in the cell, and that's the function?
So it depends on which genes are expressed in cells, which that helps to determine what the function is. Okay. What else? Yep.
Any signal transfer happens that would turn something on or off inside the cell, like the moving signals.
Okay. Signal transduction. Is that what you're saying? Okay. Do you see someone else? Okay. Well, yeah. So I'm trying to start leading into these accessory proteins and how they're going to be regulated differently in different cells. Okay. So actin and tubulin are highly conserved. And so studies in yeast show that actin interacts with many different proteins involved in many different processes. What does it mean to be highly conserved? What does conserved mean? I say that a lot, so hopefully I keep seeing the same people today. Yeah. Oh wait, yeah. Wait, I'm gonna go behind you because I heard you once. Yeah.
Type, like, tightly regulated.
Regulated, not quite.
Same or very
The
similar?
same or very similar. Okay, so you things can be conserved among many different species. Right? Yep.
Does it mean that like they haven't changed
They haven't changed over. They haven't millions and millions of years. Okay, yep. So they're conserved. They're the same among species. They're yep. No. Yep.
They have the same sequence, same motif.
Same sequence, same motifs. Okay. We're getting there. Yep. What else? So what might this? Yep.
I just say that they play such an important role that they're constantly selected for.
They play. So they play an important role. So they're selected for, meaning they need to maintain the same sequence because of what? Yeah.
They're found in pretty much every cell type, so because they're used in every cell type.
They're found in every cell type. They're used in every cell type. They have the same sequence. So a sequence of a protein often has something to do with the proteins it's going to interact with, right? So if actin and tubulin are highly conserved, it kind of makes sense because they're involved in a lot of different cell processes. They have a bunch of different proteins that interact with them in order to help regulate them. Good. Okay, so let's go away from actin, tubulin for a minute. Actin and tubulin are common to all eukaryotic cells. Intermediate filaments are only present in vertebrates, nematodes, and mollusks. So they're not required in all cell types, only in cells subject to mechanical stress in squishy animals. So we spoke about this briefly the other day. You're not going to find intermediate filaments probably in insects or in organisms with cell walls. Probably not. They're constructed from subunits made of monomers that dimerize and associate with other dimers to form a staggered tetramer. And then those tetramers are going to pack together. No NTP binding, so no ATP, no GTP, no polarity. So they're not going to have the same kind of dynamic instability. The 8 tetramers are going to pack together to form a rope-like filament, which is easily bent and difficult to break, which kind of makes sense when you think that intermediate filaments are important for maintaining the structure of cells in the tissue, for example. So they're just like there's many types of actin and tubulin in different cells or in different cellular locations. There are different types of intermediate filaments with different functions. And so there's nuclear, lamentin-like, epithelial, axonal. My favorite hated are keratins. Anyone who's done any, who's run any SDS page gels and tried to stain it or do any sort of mass spectrometry to identify proteins know that keratins are all over you, and all you have to do is touch your gel, and then it's a mess, basically. They're all over everything. So we hate keratins. But they're important. So they provide mechanical stability. For example, keratin, my favorite hated. There are 20 types of keratin proteins found in human cells. So here keratins are stained. You can't really see the nuclei. Actually, no, those are microtubules. Keratins are, I think, are all around the edges. They're cell type specific, like I just said. And the diversity of keratins is very useful clinically in the diagnosis of epithelial cancers because since different keratins originate in different cell types, you can determine the origin of a cancer. So that's useful. They provide mechanical strength, and they anchor. So there are anchor intermediate filaments. And you can see they basically are at sites of cell-cell contact, and you can see them in blue here, called desmosomes, and at sites of cell matrix contact, so called hemidesmosomes. And if you have a defect in an intermediate filament such as keratin, then you're going to have, there are medical issues associated with such as blistering. You'll get detachment of your epithelial cells from your basal lamina. So this is showing mutations in keratin, and if they're involved in a number of diseases. This is normal. This is a cross-section of the skin from normal tissue, and then here you have a defect, a mutation in keratin, which results in this blistering called epidermolysis bullosa simplex. Easy, rolls right off the tongue. And so this happens when defective keratins are expressed in the basal layer of the epidermis. ALS and Lou Gehrig's disease is associated with accumulation and abnormal assembly of neurofilaments. And so these are intermediate filaments that are found along axons and vertebrate neurons, and the defect may interfere with normal axonal transport. Actin and tubulin are also frequent targets of toxins produced by plants, fungi, and sponges, and this is actually useful. Well, it's not useful if you encounter these things, just when you don't want to, but they're useful in the sense that they give us tools for research in the lab. For example, latrunculin from the sea sponge, it binds actin monomers and prevents assembly. What do you think that does? What do you think the result of that is? What happens if you have all your actin monomer bound and you can't polymerize that into actin filaments? What do you think? How might that affect the cell and the function of the cell? Did I hear someone say it's not good? Yeah. Right. Would it not be able to move? Who thinks the cell wouldn't be able to move? That might affect cell movement because cell movement is, you need actin to polymerize for the cell to move. Right. So that is a useful tool in the lab if you need something that's going to prevent actin polymerization. Then there's phalloidin, and that binds to and stabilizes actin filaments. Again, is that good for the cell? Remember, we need this dynamic instability. This property of dynamic instability is important for the cell's ability to respond to the environment, right? And so if you have some protein that's going to bind to and stabilize actin filaments so that they cannot disassemble, what do you think might happen? So with the cell, the cell can't move because you need that ability to assemble and disassemble and the treadmilling properties for movement to occur. Right. And then there's colchicine, which is from the autumn crocus. That sounds so nice. But it binds to and stabilizes free tubulin. What do you think the outcome of binding and stabilizing free tubulin might be? What do microtubules do? Yes?
I'd say that organelles wouldn't be able to move, molecules wouldn't be able to move around them, and then the cell wouldn't be able to divide during mitosis.
So right, organelles wouldn't be able to move. Cell division, organelles wouldn't be able to move. Let's think about that for a minute. Cell division wouldn't occur, so that might inhibit your mitotic spindle. Let's think about organelle movement for a minute. Yes?
This is mildly off topic, but colchicine is something, I'm sorry. Colchicine is something that they use for gout, to treat gout. And I think it's like, it prevents like inflammation or something like that. It helps to get the crystals out of your joints and stuff. So is it, would it stop the crystals or something?
I don't know the mechanism. So it's used for treatment of gout. You're saying? Who's taken colchicine before? Actually, I once had a student who had taken colchicine. So is it an anti-inflammatory? I need to look this up. Well, if it's an anti-inflammatory, it would make sense because if it's stopping cell division and it's stopping expansion of the immune cells, that would decrease
All I know is
inflammation.
you're supposed to take them up and it makes you sick. So
Okay,
you get rid of everything.
You're supposed to take, and that's what makes you sick.
Flush everything.
So let's just go back to organelle movement. Sorry. It's okay. It's okay. I like digressions. So let's go back to organelle movement for a minute. If you treat yourselves with colchicine and you're stabilizing free tubulin, would your organelles be able to move? I want to say you're right, no, because you're not going to be able to grow your microtubules. But then there's a part of me that says, you're probably right. So where my brain is going is thinking about motor proteins, which we haven't spoken about yet. But really the organelle movement has to do with motor proteins. And so it might not, but you're probably right. Okay. We'll go back to gout.
Good.
I'm going to look it up. Okay, so Taxol is from the bark of the Pacific yew tree, and it binds and stabilizes microtubules. So again, what's the effect going to be on cell division? It's going to, if it inhibits, So if it inhibits microtubules, then you're going to not be able to divide your, right? You're not going to be able to do chromosome segregation. You're going to have a completely non-functional mitotic spindle, right? And so it's used to treat breast cancer and lung cancer, and it preferentially kills dividing cells, which again, does this make sense? Dividing cells are busy using their microtubules for cell division, so it makes sense. So this is just a picture of HeLa cells at metaphase. So you've got your microtubules are in red, and your chromosomes are in blue, dappy. DNA. And the cell's treated with Taxol. And so, in this case, your chromosomes are failing to attach. You can see a chromosome down here. You can see one over here. They're failing to attach to mitotic spindles appropriately, and that results in cytokinesis failure. But either way, still, if you're binding and stabilizing microtubules, it's catastrophe in part that causes chromosome segregation. So if you can't disassemble your microtubules, then you can't segregate your chromosomes. But aside from that, you still are not, so we'll get to it when we learn about mitosis, but you're having this kind of probing of your kinetochore spindles for your kinetochores of your chromosomes. And if they're not able to do that, they can't properly attach to the chromosomes. Okay, so this I thought was very cool. So I never really, well, I don't think about bacteria very often, which is unfortunate because there are more of them than there are of us, right? So they have a tubulin homologue, which is called FTSC. So I just put this stuff in here because it's fun. This is what cell biologists do for fun. So it has a tubulin homologue, and its structure is similar to alpha beta tubulin, and it also binds GTP and it's hydrolyzed when microtubules, when the monomer polymerizes. And it is also dynamic with a half-life of about 30 seconds. But it actually functions completely differently in a way from how tubulin functions, or it has a different job from tubulin in eukaryotic cells. So this FTSD forms the Z ring of the septum during cell division. And so I know we haven't yet spoken about mitosis and cytokinesis, but does anyone know what cytoskeletal protein is important for the equivalent of the Z ring? We call it the contractile ring. No? Actin forms a contractile ring. So in bacteria, it's actually the tubulin homologue that's forming a contractile ring-like machinery. So it rearranges rapidly during the bacterial cell cycle. So you can see it here, and then you're about to divide, and so you have this movement of the FTSZ into what's going to be the septum. And so it's involved in pinching the cell in two after chromosome segregation, which is similar to the contractile ring. It's also, this homologue is also in red algae and is involved in dividing chloroplasts. Again, similar to the contractile ring, but the contractile ring is made from actin. Bacteria also have homologues of actin called MREB and MBL, and they play a role in cell shape and chromosome segregation. Again, kind of different because these are homologues of actin. And when we think about chromosome segregation, we're thinking about what cytoskeletal protein? Actin? What cytoskeletal protein do we think about when we think about chromosome segregation? Tubulin, right. So, different. These are bacteria that have wild type MBL, and these are ones that are deficient. And so you can see with wild type you have your normal cell shape, and they have this kind of curved shape when they're MBL deficient, suggesting that this actin homologue plays a role in cell shape. And that kind of makes sense because actin also plays a role in cell shape in eukaryotic cells. So similarities and differences, fun things. So then there's a bacterial actin homologue called PAR-M that's responsible for plasmid segregation. And so it exists in monomers and polymers also. And it will bind to the origin of replication. And rather than in eukaryotic cells where you have the separation of your sister chromatids, here it actually, well, let me change that. Here it actually polymerizes and pushes. It's actually pushing the bacterial plasmids, the bacterial genome across. So then there's another homolog in bacteria. This is an intermediate filament homolog called crescentin, which is responsible for the crescent-like shape of this Caulobacter crescentus. And so here it's stained here in red. And so it's involved with this curve. And so here you've got your normal bacteria, which express crescentin. And then if you delete that gene from the genome, then you've got this straight bacteria. So suggesting it's important for the shape. How are we doing? Okay. Okay. So what's the relevance of presence of homologs of actin and tubulin in bacteria? Why am I telling you about this? I think it's fun, other than it being fun. It's nice to think about similarities and differences. You know I love to think about similarities and differences between everything I teach you. So how are their functions similar to their eukaryotic homologs? How are they different? What function is missing? So we've seen functions in shape. We've seen functions in plasmid segregation, in cell division. What function is missing? I told you about tubulin homologs and actin homologs. What function is missing from what I just showed you? Yeah. We didn't really talk about the role of actin in movement. Yeah, we talked about it in cell shape, but not really in movement. Yeah, that's one. What's another one? Not to say that it doesn't exist, but not that I know of. Is there another one? What did we talk about with microtubules? What's an important role for microtubules? Yes?
Organelle movement.
Organelle movement. Yeah, so we didn't really see any evidence of actin. Well, actually, we did talk a little bit about chloroplast.
That's division.
But division, really. It's just division, yeah. So we didn't really see any organelle movement, which kind of makes sense. Good. Okay. So the cell needs to regulate the length and stability of its cytoskeletal filament. I keep talking about this dynamic instability, but is that very helpful if there's just this random growing and shrinking, growing and shrinking? I mean, I guess it's partially helpful, but it's nice. Remember, I'm really big on regulation and how nothing really happens randomly in the cell, right? And so all of these accessory proteins exist specifically to regulate the length and stability of these cytoskeletal filaments. So I want you guys to look in your textbook because there's a very nice figure, panel 16.3 in Alberts, and it's very nice because it shows actin filaments and microtubules, and it shows all the different accessory proteins and how they bind and what they do. And so I want you guys to do this on your own anyways as we learn about them so you can start to think about where everything comes into play. But this is a good place to look if you're confused on where to start. Okay, so we're going to start with proteins that nucleate elements. Okay, so what is nucleation? Okay. Chelsea Silver. Where is she? Where is she? Okay, Chelsea's not here. Nina. What is nucleation? And feel free to talk to a friend. So right, the binding, that there's that constant binding and unbinding of actin subunits or tubulin subunits until they reach this stable aggregate, and then they can start growing, right? So how does this regulate microtubules? How does nucleation regulate microtubules? Do you have growth or disassembly? Growth. So when you have so if you nucleate your microtubules, you're going to encourage growth. And in actin, the same thing. You're going to encourage growth. So gamma tubulin is involved in nucleation of microtubules as part of the gamma tubulin ring complex. And so here it's showing your gamma tubulin, and there are some accessory proteins that are involved in forming the gamma tubulin ring complex. And what it does is it acts kind of as a nucleus and binds to subunits to help to encourage growth. So remember when we spoke about an eraser. So remember when we spoke about just the dynamics of actin polymerization or microtubule polymerization. It kind of looks like a bacterial growth curve a little bit, right? And you've got this lag phase. And so that's a period of time that it takes to form your nucleus. And then once your nucleus is formed, so you've got this on and off thing. And then once you form your nucleus, which is that stable aggregate, then you go into your exponential growth phase, which is where you make a very long microtubule until you hit your steady state, which is at your critical concentration. And then so this would be where you have the equal on and off, right? Off, on. And that's your critical concentration, right? So what your nucleus is doing is it's bypassing your lag phase. And so you're able to start right here and immediately grow your microtubules or your actin. And so gamma tubulin and the gamma tubulin ring complex is a nucleating protein or nucleating complex that encourages growth of microtubules. So in animals, a microtubule organizing center, which I'll often just write as MTOC, it's located near the nucleus, and it's called the centrosome, and it contains more than 50 gamma tubulin ring complexes. And so that's why when you look at pictures of the cell, and you look at pictures of the cell in interphase, the microtubules seem to kind of be emanating from the nucleus. They're not truly emanating from the nucleus, but your centrosome is right near the nucleus, and it contains all of these gamma tubulin ring complexes, which are nucleating microtubule polymerization. So you have your microtubules growing out of there. So this is where microtubules are organizing, and they're pushing outwards against the cell membrane, and that's what centers the centrosome. So you can imagine if you have something in the middle and it's constantly pushing against the cell membrane, it's going to center itself. And so during interphase, more during prophase, pro-metaphase, they duplicate and move to opposite sides of the nucleus to form your mitotic spindle. Did everyone already know this? Oh my god. I guess I'm done. I thought I had more. Okay, so you guys get to go a few minutes early. I will have exams at recitation.